So it's my pleasure to introduce Rob Beiko for giving the last talk in 2016. This is his fourth talk in this single trip to Vancouver. So he was here for a project meeting that we had for the last 3 days and gave 3 talks in a single meeting. But anyway, I'm here to talk about Rob, not about his talks. So Rob did his PhD at University of Ottawa and postdoc at University of Queensland in Australia, broadly focused in analysis of microbial genomics and metagenomics. He's also a Canada Research Chair in Bioinformatics. Okay, so it's pretty broad. And he's been at Dalhousie since 2006 and is currently the full professor in computer science. And you have another department that you're familiar with, right?
So mathematics and
statistics. Okay, and mathematics and statistics. And he's also contributing to the sort of bioinformatics community in Canada through his involvement in Canadian bioinformatics workshop training and also in sort of a steering committee for computational biology and bioinformatics in Canada. So today he's going to talk about a coevolution-based approach to the identification of genes with similar phylogenetic distributions.
Just making sure. Is there like a presentatic that you want to do on the next slides?
There's a camera aiming at you, so you might not wonder too much.
So Will mentioned the Canadian Bioinformatics workshops. So I don't know if he's mentioned it to you, but he's actually organized a new one in genomic epidemiology last year. Did you know that? Okay. He's leading it. Awesome. And it's going to be in Vancouver, of course. So you should totally go, whether or not you do genomic epidemiology. So so I appreciate those of you coming in spite of the title. It's very long and very kind of boring, but I really enjoy putting this title and this talk together because it's kind of taking me back to my roots a bit in microbial genomics and lateral gene transfer. I've been pulled in so many directions that it's really nice to come back to this evolution in a very, very different context. So So the student who's been doing this work is Chagrad Liu, who is he was a master's student and then he graduated from that, and then he decided to stick around and do a PhD. And this is just to demonstrate to you that, yes, it is as cold, if not colder, on the other coast as it is here. Hong Gu, who did send a picture last night, is the other, she's the co-supervisor, so she's in mathematics and statistics. She provides the mathematics and statistics credibility to this project. All right, so here's what I'm going to tell you about today. I'm going to talk a little bit about lateral gene transfer, and then give you sort of a sense of what phylogenetic profiles are and what we might be able to do with them. And then evolution is a thing, and we need to think about it, especially in this context. And then just briefly going to touch on the relationship, the somewhat tortuous and sometimes embarrassing relationship between homology and function. Sometimes they're treated as the same thing, but they're not. And then results, and then what comes next. All right. So, lateral gene transfer, you know what it is? Yes? No? Okay, so usually we're used to thinking about genes being passed from parent to offspring, mutation, duplication, yada yada. But bacteria and archaea, and to a lesser extent some interesting eukaryotes, like to share genes back and forth. I shouldn't call it sharing because often the sharing involves the donor exploding and its DNA being released. But the basic idea is that microorganisms, can pick up genes from their environment. There are mobile elements like plasmids that are good at moving genes from one organism to another. Often this transfer happens between closely related genomes, right, from E. coli to E. coli, but sometimes it happens at very, very crazy distances. So this is a useful thing to think about, and we cannot really understand microbial evolution without considering LGT, which is pretty much a central process as microbes evolve and adapt and deal with new situations and grab new opportunities. And so we've done some work over the years looking at what I call highways of gene sharing. The basic idea here is that gene sharing, lateral gene transfer, LGT, is not random between organisms. If you take a set of organisms at whatever taxonomic resolution you want, there are going to be certain sets of organisms that really like to share genes with each other or have historically shared a lot of genes with each other. And this is just one example I'm showing you of different classes of bacteria. I mean, this is really, really broad. This is like almost the entire diversity of bacteria you're looking at here. And the idea is that we've got these different classes, and the heat map shows you the relative tendency, or if you build phylogenetic trees, how often do we see, let's say, Deferribacteres together with Synergistides, for example. And so you can see that it's not quite random, and there are some groups that actually do really, really, really like to share with each other relative to others. So white is kind of like, never mind, I'm not talking to you. At the other end, red is, they get along really well. And you can turn that into a network on the other side, and you can see that there are some really strong connections, so there's the big black edges between certain groups. And then there are other groups which have basically nothing at all, or not enough to deserve getting an edge between them. So there's non-random sharing here, and what's going on there. I showed you classes. We can look at this at the genus level as well. So it's the exact same thing. It's a heat map, right? We love heat maps. And you can see that, again, there's some non-random sharing between specific genera. And you can look over on the left. So the coloring of the network is by phylum or class. And you can see some really strong connections within groups, which is not surprising. But also you can see some preferential connections among groups. In case you're curious, you can see that big yellow dot there with like the mohawk of connections to other yellow dots. That's Clostridium, in case there are any Clostridium fans out there. All right, so why is there this preferential sharing? There are a few reasons for this. Some of them are mechanistic, and some of them are more kind of evolutionarily interesting, i.e. adaptive. And so one, and I didn't know this word before I got into lateral gene transfer, propinquity, which is basically just if you live in the same place, you've got bacteria around you exploding all the time, DNA released into the environment. So that's what you're more likely to take up. There's this constant rain of DNA falling on you. Some of it's going to end up in your genome. A lot of it's going to get swept out of your genome, but not always. Okay, so that's one factor. Another factor is the predisposition to share genetic material. And this often comes about because different bacteria might have similar transfer mechanisms, right? There's a plasmid that can infect this and that. Also, there are factors like nucleotide composition. It's been shown quite a few times that if our genome compositions are similar, I've got a high proportion of GC and you have a high proportion of GC, then we have a much higher tendency to kind of accept each other's DNA because it's easier for it to recombine into the genome. And then the one that's perhaps the most interesting, or the most relevant to this talk, whether or not you think it's interesting is another matter entirely, is potentially the adaptive sense, right? And we have to be careful when we use the word adaptation. But the basic idea here is that you might have a set of genes that give you a certain advantage in a certain habitat, right? We're all drowning in, I don't know, glucose, and you've got genes to metabolize glucose. Wouldn't it be great if I had those genes too? And so processes happen, right? And I acquire those genes, and I'm able to metabolize that as well. Antimicrobial resistance is another great example, right? If we're bathed in antibiotics, either you have certain resistance attributes or you're dead. Therefore, if I acquire the right genes, I can be not dead and contribute to the next generation of bacteria. Right? So again, there's this sort of selective aspect to this. And we see this a lot. We see that genes we might call adaptive are shared across wide evolutionary distances. And so turning things on their head a little bit, you say, here's a bunch of genes that have common evolutionary histories. You say, well, why? Right? If we see a bunch of genes that look like they've been transferred from Clostridium to, I don't know, what's your favorite bacterial genus, Pseudomonas, right? What is it about those genes? Does that set of genes, let's say 50 genes, do they share anything in common? And, well, long story short, sometimes they do and sometimes they don't. But it's interesting if they do, because that might tell us something about the environment, might tell us something about the ecological context of these things. So we do want to understand this. Now, one tricky problem is that it's not as simple as saying, here are two homologous sets of genes that are found in exactly the same sets of genomes, right? Think of having 100 genomes, and gene 1 is yes, yes, no, yes, yes, blah, 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 blah. And gene 2 is yes, yes, no, yes, yes, equivalent, blah, 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 right? There are quite a few reasons why you might have these interesting patterns in common, but they're not perfect. One reason, if you've ever dealt with next-gen sequence data, you know very well that the genomes you get are incomplete. Sometimes there are incorrect assemblies, and some of the genes you're most interested in will be bye-byes, right? They will not be there, okay? So that's one reason. You might have a gene that looks like it's missing, when in fact it isn't. So this is obviously going to lead to distributional differences. Another reason is that, maybe you've got the right assembly, lucky you, but one of the genes has recently been lost from a subset of the genomes, right? So selective meh is basically, these used to be important and useful, but now, not so important. And so suddenly if you've got two different genomes in the same environment and there's a set of genes that they shared that are no longer useful, they'll tend to lose them fairly quickly, but they might lose them in different orders, right? Because there's no sort of specific pattern of loss, right? There are some really interesting genomes. Mycobacterium leprae is one, so the causative agent of leprosy. I don't know if you know, but the genome actually has leprosy because about 1,500 genes are no longer useful in its new environment. And so you can see kind of echoes, residual signals of these genes, but the genes themselves don't work. That's an extreme example, but you see this all over the place. Another possibility, another reason is simply that you can have different sort of complementary or alternative pathways to achieve the same goal, right? If you look at metabolic networks in KEGG, you can see that there's one way to get to butyrate, and there's another way to get to butyrate. Maybe you don't need both, right? And so in some cases, you might see that the common genes are present, right, in all cases. But then in some cases, you might have option A that is present in this subset of genomes and option B that's present in a subset of genomes. So again, we're going to have, hopefully, similar patterns, but not identical. So we need to figure this out. And so why is this important? Or perhaps a more refined question is, why do I find this interesting, and hopefully other people? Is that you can take a set of genomes. You can take one genome. And the bacterial genome has, 3,000, 4,000, 5,000 genes. The monster, as far as I know, is still Candidata Syringium cellulosum, which has, I think, 11 or 12,000 genes, which is like twice that of yeast and starting to get up into multicellular eukaryote territory. But no matter what the size of the genome is, you can try to come up with summaries of the function of the genome in terms of individual genes, KEGG orthology groups, pathways, whatever sort of surrogate for function you happen to fancy. But that, you can take that a long way. You can get a lot of insight from that. But think about now being able to identify these sets of genes that share common evolutionary histories. In particular, common weird evolutionary histories, such that here's the diversity of bacteria, and there's a set of genes that are found here and here and here and here and nowhere in between. If you can find many genes that have that kind of pattern, then that suggests strongly that they have something to do with each other. And so instead of just saying you have these 25 genes, you can say, oh, these are a module that tend to show a similar phylogenetic distribution. And so think about the genome now, not as a set of several thousand genes, but as a combination of modules. If we do that, that might give us further insights into the evolution and the ecology of these organisms. So let me introduce you to phylogenetic profiles. These are not new concepts. They're approaching their 20th birthday, and they're really a product of the early age of genome sequencing. So 1998, so the work would have been done in 1996 or 97. First bacterial genome was completed and published in 1995. It was, who knows what it was? Haemophilus influenzae, exactly. E. coli was a couple years later, yeast was 1996, and so we started to get more and more complete genome sequences where you could say, yeah, right. And it was in the days of Sanger sequencing where people actually cared about closing genomes. Spent $10 million sequencing a genome, why not take a few days to close it? And so you had these presence-absence patterns, and you could suddenly say, all right, well, we have enough genomes that we can take two homologous sets of genes and compare their distributions. And so the distribution of a set of genes across a set of genomes is simply what we call a phylogenetic profile. And you can see here, each red line represents a different genome, and then the column there with the gray stuff in it is, well, there's a gray bar if a given gene is present in a certain genome, and there is no gray bar if that gene is absent from the particular genome. So this profile tells you immediately what the distribution is. Okay, so that's maybe interesting. What can we do with that? Well, we can take one profile and call it the reference, and then we can compare other profiles. So now we have 4 sets of homologous genes. We can say, here's my presence and absence pattern, here's your presence and absence pattern, here's your presence, and so which of us are closest to each other, right? And so in this case, you can see the reference, and all I've done is come up with, well, Chirvet has come up with 3 different conceptual profiles, and at the bottom we have some distance calculation between our reference and each of the other 3 genes. And so, you know, the number there is pretty obvious when you look at the difference, because there's all sorts of gray bars near the top in gene 1 that are not present in the reference gene. Are they dissimilar? Yes. Duh. Obvious. But we can quantify this, right? And so this is just an example from the days of yore, where in this paper where one of the papers introducing phylogenetic profiles, they said, here are 5 genes, right, ribosomal genes, that have identical profiles. Let's look at stuff that's one bit different. What does that mean? It means that this set of 5 is present in a certain set of genomes. The ones on the right are the same set plus or minus 1 genome, right? So the ones at the top might be, you know, the same as those 5 plus, I don't know, E. coli or Salmonella or Aquifex aeolicus or whatever, right? And so you start to see these similarities, and guess what? Ribosomal genes all over the place. On the right, a different set. Those are mostly involved in amino-acid metabolism. So again, you can see that these linkages among profiles really can start to tell you something about shared function. And so the general procedure that I'm going to walk you through is like this. First, you choose your genomes. 2, you need to identify your homologs. You need to figure out which genes in each genome are homologous with genes in other genomes. From that, you can build your phylogenetic profiles. Once you've done that, you can compute the distances among your phylogenetic profiles. And then finally, you can build clusters. So I've shown you each of these steps with the exception of the last one, because you can take all of your profiles and do all versus all comparisons and say 35, 16, 22. But at some point, you might want to have a criterion to say, I'm going to draw a line around these ones and try to say something about them. It's different from what I'm trying to say about those. Okay? So that's all fine and good. Hopefully I've convinced you that this is potentially useful. But there's a problem. So what I refer to as naive approaches, such as Manhattan distance, which is simply the number of differences, Euclidean distance, right, the square root of the sum of squares, you know, whatever magical distance you have, will ignore one very important factor. And that factor is the evolutionary correlations among your genomes. What do I mean by that? Well, this is exactly the same figure I showed you, right? Reference profile, profile 1, 2, 3, distances, but there's something I haven't shown you yet, right? Each of these rows corresponds to a genome. These genomes are related through a process called evolution, and therefore one can draw a tree relating those genomes. And guess what? that huge pile of gray bars at the top are basically from 21 copies of the same organism. Right? They're different. They were isolated separately. They were sequenced separately. But they're all different strains, in this case, of Clostridium difficile. These are real profiles. And so what does this mean? Well, it means that if I have a set of genomes, I can calculate these distances. But then imagine taking one of these genomes and running off 100 photocopies of it. You can still build these profiles, but without actually changing any of the information you have, you can actually really mess up your profiles. Right? So what do we do about this? Well, hopefully, intuitively, you can already say, well, let's do something about these relationships. And so we've tried a few things, but the best one that Chara came up with was another fairly old thing, which is a coevolutionary model. This is based on a method developed by Mark Pagel back in 1994. What's the key question here? The key question in Pagel's method is if we take two traits, and so in the initial paper, I don't even remember, but the traits could be, the presence or absence of certain physiological structures in charismatic megafauna. But in this case, our traits are simply genes. And so now we ask the question, do two genes evolve in a dependent or an independent fashion? If they evolve independently, then we'll probably expect kind of random distributions with respect to one another. Now, of course, the tree-based relationships will tend to give us patterns that look similar. So we have to take that into consideration, kind of subtract out the tree when we ask these questions. And so this works by considering alternative models. And it's right there. It's, do the two genes evolve dependently, or do they evolve independently? I'll show you the models in a moment, but the basic idea is that the dependent model has more parameters than the independent model. So if you take these two and calculate maximum likelihood scores on a tree, you need to do something called a likelihood ratio test to determine which one is actually the better explanation for a given pair of distributions. Question?
When you say dependent, do you mean linkage to equilibrium? Are we talking within a genome or talking between?
That's a great question. So the question was, when I talk about dependent, am I talking about linkage disequilibrium? So two things that are close to each other in the genome. And as you'll see later, it leads to some really fun terminology clashes, because I can talk about clusters and clusters. In this case, it has nothing to do with their physical location on the genome. It's all about whether they tend to be found in the same sets of genomes once we've subtracted out the influence of the tree. Now, genes that are next to each other in many genomes are more likely to have these common distributions because when they get shared, well, guess what? They're together. It's more often the case that they'll get shared. It's a chunk of DNA that's getting passed back and forth. So maybe they do have a better chance of being shared. But this, in this case, has nothing to do explicitly with the physical location. Does that make sense? Okay, great. And so we're contrasting these nested models, and then what we have for each pair of genes, instead of what I've been calling these naive scores, the Manhattan or the Euclidean distance, now we have a test statistic, which basically says if the test statistic is large, then we probably have traits evolving in a dependent fashion, and we're more likely to want to put these two genes in the same functional cluster. If they're independent, well, forget it, right? They're off in their own separate worlds. And so these are the models, and this is as mathy as I'm going to get because I'm a biologist, not a real computer scientist, but the basic idea here is that we have our two models. In the independent model, we have 4 parameters. Okay? So we have two things that we're looking at, two traits, so we get two genes, right? And so gene 1 can go from absent to present, or present to absent, right, as we think about its process along a phylogenetic tree. And so we can have two parameters, right? So trait A, there's a parameter that describes its probability of going from 0 to 1, which means absent to present, to be present through lateral gene transfer. and then the reverse. Okay, so we have separate parameters, right? Gene A has a certain probability of being gained and a certain probability of being lost. Gene B, same thing. Probability of being gained, probability of being lost. So when you see beta 1, beta 2, alpha 1, alpha 2, that's exactly what's being described. The key here is that gene 1, when it's being gained or lost, doesn't care about the state of gene B. Right? It's totally independent. That's the independent model. Whereas down here, notice that I've got two parameters, Q12, Q34, right? And in both cases, character B is going from state 0 to state 1. So gene B is being gained. But because there's a dependency, I need different parameters for these two. Because now gene B actually cares whether A is present or not. So A is influencing the probability of B being gained or lost. So below, we have the more complex model. Now, if you've ever dealt with the pain/fun of likelihood, you know that a model with more parameters can trivially be fit as well as a model with fewer parameters, right? Because the bottom model can reduce to the top one. And what's more, usually with a model with more parameters, it'll find ways to tweak the values a little bit so that it kind of pretends to look better even when it's not. And so the likelihood ratio corrects for this because we have more degrees of freedom on the bottom. So likelihood ratio test says, okay, well, we're going to fix that. And the degrees of freedom is simply the difference in the number of parameters, and it's chi-squared. Okay? So that's your distribution. So these are the hypotheses that we're comparing for each pair of genes. And so what does this mean in practice? Well, it means that instead of treating these independently, the tree-based model can look at the tree and say, no, that is one gain event. It's not 21 or 23 independent things, it's one. And so Pagel's method will properly account for this and downweight that. So this is a much more evolutionary perspective on phylogenetic profiles. What does this mean? Well, here is the difference distribution for PR to P1, P2, P3. I already showed you this. This is a Manhattan distance. And this is the dissimilarity that we calculate from Pagel's method. So notice that P1 is still the most dissimilar. But it's a lot less the most dissimilar than it was before. What's more than that, the ranking of P2 and P3 has flipped. So we've actually changed the relative, we've changed the interpretation of these similarity values. And this is an important thing to do, because as I said, if we don't do this, we could take certain sets of genomes and just introduce more copies of them and get a really biased result. Whereas Pagel's method's like, no, not going to be fooled by that. And so Pagel actually did this in 2005 and showed quite clearly that it is better when you take that correlated evolution into account. And the two examples that we have, so the paired example, so there's a tree of fungi, plus a couple of non-fungal things as outgroups. And there are two pairs, right? There's the red pair and the blue pair. So each of those is a pair of genes. The zeros and ones are their distributions, presence and absence, right? And these are two examples they pulled out. And in both cases, they found differences, right? So they said, okay, in the first example, the naive approach said, oh, these are very similar, right? And if you look at it, well, yes, they are very similar, but you can explain this with a single event, right? And so the model-based approach, the Pagel model, this likelihood ratio, said, I'm not so impressed, right? And so in this case, it's kind of easy to see that it's not really that impressive a distribution when you can explain it so well in a tree. The blue example is the flip side, right? Where the naive approach, you look at that and you look at the bitwise differences, and there are not that many. But because of the way they're distributed through the tree, the model-based approach says, wait a minute, these are several independent events, so this is actually pretty unlikely. I should ask how much time I have. 5 minutes?
Yeah, at least 15 minutes.
At least 15 minutes, okay, cool. And so when they evaluated these, they evaluated a ton of them And I'm not going to show you the figures because they take half an hour to explain. Long story short, the functional similarity was better approximated with the model-based approach than with the naive approach. So that's pretty good. However, and this is where we come in, the organisms they looked at did not really have much lateral gene transfer going on. When you see these distributions in the organisms they looked at, it's usually because the ancestors had it, and then it was kind of like loss, 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 loss. It's a Dollo parsimony model, if you're familiar with that, which is basically saying things can arise once in the tree, and then they can go away many times, which for bacteria is not the case. You can have gains all over the place due to lateral gene transfer. They also used p-value thresholds. So like I said, there's this chi-square distribution of the likelihood ratio statistic, and so they used that statistic, they calculated a p-value, and then they thresholded it. That's how I was able to say, oh, this one's significant, this one's not significant. So who else hates p-value thresholds? Okay, well, not enough of you, but never mind. We use the test statistic instead of the p-value threshold. And furthermore, they looked at these relationships on a pairwise basis. So for every pair of genes, you have a test statistic and you have some functional measure of similarity. And they correlated, they associated really well. But if you want to identify sets, how do you go about doing that? That is something they did not do. And I've shown you a perfectly simple example here of the non-transitivity of these relationships. P1, profile 1, is very similar to profile 2 according to some criterion. So maybe that satisfies a p-value threshold. Ditto for P2 and P3. However, if you compare P1 with P3, not so much. Right? I mean, this happens all the time. And so what do you do about this? Right? You need some criterion to deal with this very simple situation that you can map up to the actual complexity of having thousands of pairwise comparisons of thousands of genes, which gives you like millions of comparisons instead of 3. So these are the problems we needed to deal with. A couple of caveats. This is a little interlude. Homology and function are not the same. Often, not always, often there's a conflation of the two, right? You say, okay, we've inferred homologous sets, or even more stringently, orthologous sets, right? So sets of genes that have diverged through speciation events. You can think of it as the same gene in different organisms. That's usually the way I think about it. And so often there's an assumption that orthologs should have the same function in those genomes. Wrong. Often they do, but not always. What does this mean? It means that if we use function alone as a criterion to test our results, we're not getting the whole picture, right? Because the function can sometimes be different, but we've still done a good job of looking at the homologs. Okay? Another thing is that if we find genes with similarly weird distributions, well, that's interesting. But bacteria do sometimes inherit genes vertically. In fact, probably most of the time they do. What does this mean? It means that if you find sets of genes with more boring distributions, like the common ancestor had it and all of its descendants have it, well, it doesn't mean that everything is going to have the same function, right? That could be ribosomal proteins, certain metabolic genes, cell wall, and all that. So seeing things in the same cluster doesn't guarantee that they're going to be functionally the same. Hopefully the weird distributions do show that, but the less interesting ones, maybe not so much. The other thing is the excuse, which is that there is no single right answer. There are degrees of similarity. If you think about having a graph of connected proteins, right, so this sort of, each node is a protein and edges connect the pairs of proteins, and the edge weight is the likelihood statistic, then you could have different thresholds. You could say, ooh, I want really big clusters, or I want really small clusters. And neither of those is going to be perfectly right, and neither of those is going to be perfectly wrong, because you can have degrees of similarity. Right? So we need to keep that in mind. So let's get on with it. That's the procedure again. Genomes, homologs, profiles, profile distances, build clusters. Part 6, interpret clusters. So let me introduce you to one of my favorite bacterial friends. Lactanospiraceae bacterium 3157 FAA CT1, okay, which we affectionately call Lachnozilla. All right. So this is a bacterium we're kind of interested in, and I should say this, Emma Allen Verco in Guelph, she's at University of Guelph, isolated this from a fecal sample, of course, from a patient with Crohn's disease. Does this mean this bacterium has anything to do with Crohn's disease? Of course not. Maybe it does, maybe it doesn't. We don't really care. Well, we care, but that's not our primary motivator here. Why do we care? Well, two reasons. One is that we're really interested in the Lactanospiraceae. Why is that? Because if you look at samples of the human microbiome, you see a lot of Lactanospiraceae, right? So this is a family of bacteria. And within this are many different genera, Anerostipes, Stipes, Blautia, Butyrivibrio, yada yada, all the way down. Lactobacterium, very creative. So all of these, and a lot of them appear to have constructive roles. Constructive in the sense of they synthesize molecules that make us happy. Constructive in the sense of they can actually, seems like they can be protective against certain pathogens. So they're interesting to study for that reason. And why Lachnozilla in particular? Why is it called Zilla? Because its genome is really big by bacterial standards. Okay, remember this is the game we're playing here. So this is all of the complete assembly and draft genomes from class Clostridia. Okay, there's a lot because it's a really important class. And all you see is that each row is a genome, and the column, well, this tells you the size of the genome. So the smallest ones check in at around 1,000 genes. And you can see that the mode median might be somewhere in the 3,000 to 3,500 range, typical for bacteria, but that there's this little sort of subset that get really, really large. And our friend Lachnozilla is one of the biggest. It has almost 7,000 genes. So in a group that tends towards sort of a middling size distribution, what is about these giants, right? Oh, hey, look, Clostridium difficile, and Hathaway and Bolte, and so what is it about Lachnozilla that makes it so big? Right? Why is it, you know, is there some ecological reason? And just so, well, I'll disappoint you now, so you're not disappointed later. Well, you might still be disappointed, but anyway, we don't have an answer to this. But Lachnozilla is interesting because it's like twice the size of so many other Clostridia. What does this mean? Well, this is something that we're trying to get a handle on. Okay, so there's Lachnozilla for you. Now, identify homologs. We have a total of, so we have Lachnozilla, we have 687 other genomes from the same class. So in this case, Lachnozilla is our query genome. We compare against everything else. Okay? And because we like to root trees, we take 8 outgroup genomes from other phyla. A couple of proteobacteria, a couple of things from class Bacilli, which is kind of Clostridia's neighboring class, and we do the homology search, Lachnozilla versus all of these things. Algorithm called RAP Search 2, which is basically as good as BLAST, but a whole lot faster. And then we set an e-value threshold. Okay? Nothing too magical. Step 3, build the phylogenetic profiles. And so under this search criterion, you can have a gene from Lachnozilla, or a predicted protein from Lachnozilla, that matches several genes, or proteins, in one other organism. And long story short, we don't really care how many proteins it hits. As long as it hits one, we set the value to 1. Okay, so it's a binary string, it's a bit string, present or absent. Okay? There are many other ways you could do it, but this is our starting point. Okay, so those are our phylogenetic profiles, right? 1s and 0s across 695 genomes. And then we build a tree, because we need a reference tree for all these genomes, upon which we can apply Pagel's method. Now, if you know anything about bacterial genomics, if you've been part of these arguments, you know that you can build a tree and everybody's going to disagree with it. So this is true for us as well, but keep in mind that this tree is not a statement about the evolution of the entire group. Because we've done a lot of work and we've kind of convinced ourselves that it's impossible to get that. Is it right? Is it wrong? We don't care. All we want is some sort of rough guide to provide the correction for our phylogenetic profiles. That's what matters. And so we use this software package called Amphora2 from 2012, looked at a concatenation of 31 core proteins. And these are pretty much what you might guess, ribosomal proteins, a few other bits and pieces. XML to build our trees. And here's something that we had to do as kind of a compromise. We have the big tree from a whole bunch of genomes, but it turns out that doing these model contrasts for all pairs of profiles from 695 genomes, well, if we had done that, I would be giving this talk in about 300 years. We needed to consider a subset of genomes, and so we narrowed it down to 74, which were kind of Lachnozilla and some of its neighbors. Once we make these inferences from those profiles, we can map those back into the full set of genomes, but I just wanted to make that clear. Part 4. We've got our profiles. Let's compute the distances. I already explained how to do this, right? Likelihood ratio statistics. Now, the likelihood ratio statistic is an expression of similarity. Okay, so we need to convert that to dissimilarity, to distances. And the very sophisticated and complex way we do that is to identify the maximum likelihood ratio in our entire set and subtract everything else from that. Okay, so if you're big, you become small. If you're small, you become big. Finally, all right, we got our pairwise things. We got some massive conceptual network of these things. Now we want to actually build clusters. We want to draw lines around things and say, yes, this is a set of genes that we're really interested in considering as a candidate module. And so Xiaoyue actually tried a whole bunch of different clustering methods, and you might see your favorite ones up there. Markov clustering is often used to identify sets of orthologous proteins, which I do not endorse at all. DB-scan is another widely used clustering algorithm. I don't really have time to get into them. But we tried the Pagel scores, the same distance values with those, and they stunk. They were terrible. Because each of these, you can apply different thresholds or different clustering criteria. And by sliding the criterion, we could get one of two answers. Basically, at one end, everything's in one cluster. Not so useful. At the other end, everything is in its own cluster of size 1. Also not very useful. So we tried those. We have figures. I won't show them to you. The two that matter most, Xiaoyue tried hierarchical clustering, which is basically, it's really simple. It says we start with each profile as an independent unit, and then we look at similar profiles, and we start to cluster them. So, you know, you and I are really similar, so we're going to merge quickly. We use the word clustering criterion, which is one way of deciding which things should be put together. And then, ultimately, we get our clusters, and we can try to draw a line at some level, and that line is basically like a hacksaw, right? We're kind of cutting the cluster trees. And what we end up with is, in this case, 3 separate modules, right? A with B, C by its lonesome, and then D, E, and F together. Those are the things that we want to look at in detail. Now, the last example, and I forgot to give the citation. It's really important because most of what I've shown you is 20 years old, right? Climb was published 2 years ago in Cell and is a method basically to do this, which is just, you know, the reason I mention this is because it's still a challenging problem to deal with. This was not solved 10 years ago. And so Climb has a similar principle. It's also phylogenetic in nature. And it's actually really cool. It's complicated, but it's really cool. The basic idea with Climb is that it first tries to identify canonical patterns of presence and absence, again, phylogenetically corrected. Okay? So it's like, here's one general model of presence and absence. Here's another one. Here's another one. And then it uses statistics to try and recruit specific profiles to those different ones. And those are its clusters. Now, a couple things about Climb. One is that it is really designed to target eukaryotes. Okay? It was tested and validated entirely on eukaryotes. One of its crucial assumptions is, again, this dollar criterion that there is one gain and potentially multiple losses. Okay? And I'm not going to show you those figures either, but Climb worked pretty well, but it had some really perverse cases where this assumption just kind of made things go nutso. And there's also no adjustable threshold. So you run it, you get your results, and you can't really tweak it to say, let's make bigger clusters or smaller clusters. Okay? So the comparisons I'm going to show you, we have our, we cheat. We have our hierarchical clustering, but we try moving that line up and down. Okay? And we see what sort of results we can get. climb at one threshold only, one set of clusters. So how do we evaluate clusters, right? You use a clustering method, you get clusters, not surprisingly. It's kind of like you use a phylogenetic tree building method. Even if all the sequences are identical to each other, right, it's really a phylogenetic dot, most phylogenetic methods will still spit out a tree, right? So how do we look at our clusters and say, all right, do we trust you? Do we have any sort of confidence or whatever that you're actually useful, that you're telling us something valuable? And so since I already dumped on function, the first criterion we can use is function, right? Because again, there's this hypothesis that things with similar distributions will hopefully have something to do with each other, metabolically, structurally, whatever, right? Number two, proximity in the genome, right? And so maybe when we have these clusters, if they show high degrees of similarity, then at least in some genomes, we'll find them together in the genome. Because again, in bacteria and archaea, we often find these operon structures where things that do similar jobs are next to each other in the genome. That's part one, the functional aspect of operons, and things that are bigger than operons. Part two is a lot of the mechanisms of lateral gene transfer, they take chunks of contiguous DNA and they throw it into some other organism. Right? They don't care about gene boundaries. They're just like, oh, DNA, right? Pick up, throw. That's not completely biologically accurate, but the basic idea is that things that are close to each other are much more likely to be transferred together. So if our clusters, if a cluster has 10 genes, and oh look, the 10 genes are one after the other in Lachnozilla, well, that's a nice quasi-independent validation that, yeah, maybe we found something. The third one is our profiles are presence and absence patterns. We use the tree to correct the profiles, but notice that we're not actually using the individual gene trees. We're just saying presence and absence. So if we take those genes and we build trees from them, we can ask the question, do the trees look similar? Or is it the same set of organisms arranged in some other weird way, right? So those are three ways that we can evaluate these things. So this is just the big picture slide. Don't try to draw too much information from it. Lachnozilla, we got 6505 phylogenetic profiles, but only 2,697 are distinct. One of the reasons for this, Lachnozilla has a lot of genes that no one else has. So what's the profile? 1, 0, 0, 0, 0, 0, 0, 0, 0, 0, 0, right, 0, 0, 694 times. We have many profiles like this, so they constitute one distinct pattern. Right? So we don't need to look at that 3,000 times. And so here's the actual hierarchical clustering relationship we get. So again, things that are really similar to each other, profiles that are really similar according to Pagel's coevolution method, are going to join each other near the bottom in that black unreadable smudge. Things will join each other higher up if they are much more dissimilar from one another. Right? So you wouldn't want to cut at a height of 400 because you're going to get 1, 2, 3, 4, 5, 6, 7 clusters. And the stuff in those clusters is not necessarily going to have much to do with other things in those clusters. So we really focus our attentions down in the sort of black clumpy parts. So, as I said, one way we can evaluate is using function, and one nicely structured description of function is through Gene Ontology. Gene Ontology, for those of you who are not familiar, is simply a hierarchical way of describing different function at different levels of precision. Right? So, there's a biological process ontology. At the top of the biological process ontology is biological process. This means that the protein participates in some kind of biological process. What kind of biological process? Well, response to stimulus, cellular process, signaling, yada yada. As we go down the hierarchy, we get more and more precise. So this is a very small subset, but it illustrates a few things. One is that things can branch but also merge, okay, because it's a fairly complex set of relationships. What is the semantic similarity? It's basically if we have two profiles and each of them has a specific gene ontology class we can put on it, similarity between these two things is basically a function of how much of this hierarchy each of them share. Right? So maybe one of them is like this, and another one is that, and so they're a little bit different, but they're a whole lot similar as well. Right? So we can quantify this. What do we get? Long story short, things look reasonably good if you look in the right range. So what are we looking at here? Well, the y-axis is the gene ontology similarity score for our clusters, right? So we built a bunch of clusters. We can look within each of these clusters and say, what is the average Go similarity in all these clusters? If each cluster was perfectly homogeneous in terms of its Go score, we would have 1, right? If each cluster was random, then we would have a very, very low score. So you can see the red dots, well, the black dots with the red lines and circles around them, that's our hierarchical approach where we move that sort of hacksaw threshold up and down. So you can see that as we move the threshold further down, right, the height of 60, then the similarity is roughly 0.8. Okay. So there's a number without context. What's the context? Well, we can do a randomization test with many replicates and say, well, if we took this tree, each of those leaves of the tree has potentially a gene ontology label on it, right? Because each of those is a phylogenetic profile. What if we take those gene ontology labels and go and randomize them? What do we get then? Well, we get that score, about 0.3 or 0.33. So you can see that if we cut the clusters at about 100, 110, we don't do much better than random. But if we decrease that threshold, we get much more functionally tight focus clusters. Okay. Makes sense. Climb, like I said, single threshold, so its Go similarity is about 0.6. So it's not too bad, but it's actually better than that. Because if we look at the similarity, here's a cluster. What are we looking at? Well, the top is the phylogenetic tree I've been showing you. You may recognize that set of 21 C. diffs. Each row now is a profile. The labels indicate the functional category of that profile. So amino-acid fans might be very happy with this because we've got leucine, metabolic processes, histidine, valine, and so on and so forth. So there's a lot of amino-acid stuff in here. That's pretty cool. What's really nice is that these will have somewhat different gene ontology labels. What does this mean? Well, maybe the average of this is 0.7, but this 0.7 translates into something that actually makes a lot more sense, right? So perfect gene ontology is neither a realistic nor a necessary goal. This is pretty useful, okay? And if you look at the profiles, the presence and absence patterns, you can kind of squint at it and say, yeah, all right, they share a fair bit of similarity. But the key here, what really, the most evident thing that Pagel's method is doing is that things that are present in all 21 C. diff are clustering with things that are absent from all 21 C. diff, because it's one event. Okay? So let's take a closer look at this. Let me explain. We can take each of these and we can convert it to an enzyme commission number, right? A specific function that would participate in a specific pathway. And then we can map these into metabolic networks and say, well, do they tell a cohesive story? And the answer is yes. So these are enzymes that were part of that cluster that I just showed you. And look at that, they're all connected to each other. Well, there's one step that's missing, right? 2, 3, 3, 13. But everything else is quite cohesive, it's connected. So this, our method is saying this is a module, and KEGG is saying, yeah, maybe it is, right? It's a cohesive set of proteins with a weird distribution that also happens to have a lot of functional cohesion. Tryptophan synthesis, a smaller part of that, similar story. It's not perfect, but it's pretty good. Second story, flagella chemotaxis. Another example of a case where perfect gene ontology similarity is neither necessary nor desirable, because I don't know if you can read this, but near the top we have flagellum. You can also see flagellum, chemotaxis. These are different Go categories, right? So we're not going to get one, but guess what? Flagella can be used for chemotaxis. All right, motility, right? And so the phylogenetic tree at the top, the distributions, again, you can look at them and say, yeah, all right, that's pretty convincing. What can we do to evaluate these further? Well, we can look at the clustered genes. And so what I'm showing you here on the top is, of course, Lactobacillus is a draft genome. Nobody's bothered closing it. And you can see the contigs lined up there, right? So there's kind of multiple contigs. The numbers there are the coordinates, the positions along the genome. And I've drawn red dashed boxes around the locations, the chromosomal locations of the genes in this cluster. The white circles are the genes that are found in these clusters. The black circles are everything else. So at the top, the clusters are pretty convincing. If we blow this up and zoom into certain regions, well, we say, actually, these things are not really adjacent to one another. In some cases, they're adjacent, in others, they're not. But an important thing to note is that, first of all, it's a mess of paralogy in here, right? So there's a lot of gene duplications. There's a lot of homologs that you can't see from this. The other is that it's pretty much all flagellar, right? So it's actually a big, big region of the genome that is annotated, at least, as having flagellar chemotaxis type things. Last one. Phylogenetic trees. Okay, so each of those profiles that we have, we can take them, we can build the alignment. From the alignments, we can build phylogenetic trees. So we get a bunch of phylogenetic trees, 20 or 30, whatever, right? From this, we could ask the global question, are the trees similar to one another? But let's not worry about that. Let's focus in instead on Lachnozilla, since that was our whole starting point. So where is Lachnozilla? Well, I didn't rename it. There it is, Lachnose-Bacterium 257FAACT1. The number of that is just the GenBank GI. And it has, as sisters, a couple of things. Clostridium boltiae, 2 different strains that look pretty much identical in terms of at least this protein. And then 2 more awfully named Clostridiales bacterium, blah, blah, blah. Okay, so that's a group of 5. Let's look at the other ones. Oh, look. There's that group of 5, there's that group of 5, there's that group of 5. Almost every single profile has that group of 5. The the rest of the trees tend to, not surprisingly, have similar genomes represented, but the topology tends to be a bit different. There's another case, and this shows up in quite a few trees as well. If you see the smaller yellow rectangle there, there's another paralogous version of the gene there from Lachnozilla, Clostridium hylamone, which I've never heard of before. I still don't know what it does, but we see that set as well. So overall, the trees have similar genes in them, similar genomes represented. Topologically, not perfect, but in the region of Lachnozilla, pretty similar. You get the same pattern repeating over and over. Okay, long story short, thanks for sticking with it to the end. The clusters, certainly the ones I showed you, other ones as well, high degree of functional similarity. The summary of the Go similarity there demonstrates that. In addition, the Go similarity doesn't tell the whole story because you can have things involved in a very similar process with different annotations. We have these trade-offs between small, highly similar clusters and large general clusters. There's no magic formula. But again, if you change the threshold slightly, you might go from a larger cluster that's somewhat similar, right, sort of somewhat related processes. You go down to a smaller cluster that's much more cohesive. Which is the right one? Well, it depends on what you're trying to do. And then just specific results, right? So we had cohesion in the metabolic network, and then we had clustering in the genome, and certain consistent features in the flagellar trees. So, what next? Right. What I've shown you is that we do presence and absence, we get these neat clusters. But what if we took it a step further, and for each set of proteins, built the trees beforehand and then tried to cluster based on those? Okay. That might give us even more information. Because you can have 2 genes with the same profile, but with very different phylogenetic trees. We didn't consider that at all. We used, and those of you who've done this before, the homology stuff was dead stupid simple, right? If it wasn't in Lachnozilla, we didn't consider it. Obviously we want to, and so one of the next things we want to do is use some sort of orthology inference so that we get a perspective from all genomes, not just keyed on Lachnozilla. Pagel's method is fantastic, but it is slow. We want something that we can apply to thousands of genomes. So obviously we need heuristics, or we need to swap it out for something that's a bit less exact. Just taking it back to the original motivation, I've shown you that we can take Lachnozilla and subdivide it as sets of clusters. What I'm hoping we can get from the next step is some idea of what is Lachnozilla. Once we start building clusters that include stuff that's not Lachnozilla, we can try to tell the story of here's what it has, here's what it doesn't have. Maybe that can tell us something about its ecological role. What is it about Lachnozilla, Clostridium boltiae, and those awfully named organisms that makes them group together in the flagellar trees? I don't know, but it's probably something interesting. The end. Thanks.
Well, maybe just a quick one or two questions, given that it says a little more stuff.
So we have these clusters. I know that we thought that we probably have a similar cluster used to figure that. What do you do with that text? Like, is there a database where you're selecting these genes belong to clusters that are similar to this cutoff or description? And then we can use that.
Okay. Hypothesis. Lachnozilla has a huge genome because it can survive under different conditions in human intestine. Right? We can have, broadly speaking, happy days in the gut, unhappy days in the gut, and things that are in there, right? And various different environmental conditions, but really, homeostasis, dysbiosis, whatever. And so, one guess that I have, which isn't by no means tested, is that Lachnozilla has so many genes because it's able to cope with multiple different conditions, whereas some of the smaller ones are like, I'm happy, or I'm dead. Right? And so if we identify modules and see that they're kind of complementary cellular defense mechanisms, complementary metabolic modules, that may tell us more about the conditions under which it can survive. Now, in terms of whether these modules, I think you were kind of asking whether they deserve to be treated kind of as canonical things, right? Is this module something that's worth throwing in the database and referring back to? That's a great question. I haven't thought of. It's kind of scary to think of that because although we would hope for more stability under this model that we adapted, adding more genomes would still change the clustering by a lot. And the whole fact that you can justify cutting the hierarchical clusters here or there really suggests that you maybe shouldn't call these canonical across multiple studies, except in a very general sense. It's kind of like we do this paper where we say, here's a set, and then you do another analysis with a different set of genomes. You're like, oh, this set is actually pretty similar to that one. Just in more general terms than the specific proteins. So my instinct on spiders and telling me that probably wouldn't want to see that. There could be cases for it.
How about the differences between mutation rates or evolution rate of different organisms?
Okay. The step where it would come into play is really at the homology inference, right? And so we used a completely brain-dead e-value threshold of 10 to the minus 20. That doesn't account for genes that would evolve more rapidly or more slowly, so that if you're doing this across some reasonably large evolutionary distance, certain genes might have their profile split in two. It's kind of like if we were looking at phylum Proteobacteria and phylum Firmicutes. Genes that evolve slowly would probably be recognizable across the two phyla at that threshold and be included in a single profile. Genes that evolve more rapidly might not be. Or, given the influence of lateral gene transfer, it could be a whole lot messier than that, right? It's like Proteobacteria that have recently received genes from Clostridia, those things, those genes that have been swapped out will appear in the same profile. But proteobacterial genes, Homologs that came from their ancestor, that came from the ancestral gene. I'm doing a terrible job of this, but it's going to be messy, right? And so, absolutely, you need something more sophisticated, something more, dare I say, evolutionary for defining the orthologous sense. Because otherwise, it's very arbitrary, and some things are going to be together, and others are going to be apart. And I have no way to know that, which is which.
I have a general question. What do we gain
by making a tree with 1,000 genes or 2,000 genes, like we compare them, like what is our goal